Speaking on this topic to this audience is both an honor and a challenge. It's an honor because so many innovative algorithms for exactly this problem have been presented at the DREAM workshops over the years. And it's a challenge because DREAM and the surrounding community has in some sense moved on to bigger and better challenges. That may be in part because of a perception that the performance of these algorithms had reached a plateau, or because of a perception that they weren't having impact on practical genomic projects. So my task today is to convince you that it is possible to scale the heights above this plateau, that we can have an impact on practical genomics projects, and that there are data available to prove that these algorithms really work. I'm going to try to do that by describing to you an algorithm called net profit that was developed by Brian Haynes, Zeke Mayer, and myself. And I have to warn you that it's not very clever. The only thing that it has to recommend it is that it works really well. But I think that we have drawn some lessons from net profit that may apply beyond this particular algorithm. And those are that expression data from single transcription factor perturbations is much more powerful than data from perturbations affecting many transcription factors at once. That the most significant responders to perturbation of a transcription factor are most likely to be its direct targets. And that differential expression data can reveal direct binding as well as in vivo binding data can, and it's a heck of a lot easier to generate. Net profit works by rank ordering all possible direct TF target relations by a confidence score that combines a number from differential expression analysis and a number from co-expression analysis in what is essentially a weighted linear combination. The differential expression analysis number is the log odds that the target is differentially expressed when the transcription factor is deleted. And for microarray data, we just use the output of lemma. The co-expression analysis is a lasso regression coefficient that reflects the degree to which expression of the target gene can be predicted from the expression of the transcription factor. Saccharomyces cerevisiae is a perfect model in which to develop these types of algorithms because most of the transcription factors have been subjected to in vivo binding analysis by ChIP-seq, and strains deleted for most of the transcription factors have been subjected to expression profiling. Unfortunately, these two data sources don't agree very well. With the genes whose promoters are bound by a transcription factor and the genes whose expression changes when that transcription factor is deleted overlapping typically by about 3 to 5%. We wanted to see how net profit could do at predicting direct regulatory binding and regulation as compared to ChIP and ChIP-seq. So to do that, we used PWM models of the binding specificity of each transcription factor that were derived from protein binding microarrays. So the data are completely independent of both the expression data and the ChIP data. And we computed the potential of each gene's promoter to bind each transcription factor. And we used three levels of stringency for what counts as adequate PWM support for a predicted direct binding relationship. And what we found was that at all three levels of stringency, the net profit predictions were supported by binding potential more than the ChIP-ChIP and ChIP-seq relations were. And this is really remarkable because net profit knows nothing about promoter sequences or binding. It's using only gene expression data, whereas ChIP is designed specifically to measure binding. Since the top 4,000 net profit predictions looked good, we decided to see how deep we could dig into this list of predictions, this rank-ordered list. So we took the top 40,000 predictions. At the highest level of stringency, 9% of these predictions were supported by binding potential models, the same percentage that was supported among all 30,000 ChIP-ChIP and ChIP-seq relations found in the TNet and YeastTrack databases. So we concluded that the net profit algorithm based on expression data is able to recover more direct targets than the chip-chip experiments are at the same level of specificity. We also wanted to see if we could find new transcription factor functions with net profit that couldn't be found by chip-chip or chip-seq. So we took all the Go terms that were enriched in the net profit predicted targets of each transcription factor, and then we dropped out all those that were enriched in the chip predicted targets of each transcription factor. And we found 44 functions that were assigned by net profit but not by chip, of which 29 were supported by other data sources, primarily mutant phenotypes. 2 were supported by data from homologs in other species, and 13 were completely novel. We carried out experimental validation on 2 of those completely novel predictions and validated and verified that CBF1 amplifies the effect of inorganic phosphate on the expression of phosphatase genes, and EDS1 is a specific repressor of genes in the lysine biosynthesis pathway. And I'll point out that EDS1 had no known function before. We also compared net profit to 2 top performers in previous gene challenges, Inferolator and Genie3. And we compared them both in terms of the percent of their top 4,000 predictions that were supported by PWM binding models, the percent that were supported by chip evidence, and the percent that were supported by both. And you can see the results here. So what did we do differently from these other algorithms? First, we used a global shrinkage parameter for lasso, 1 shrinkage parameter to cover all genes, whereas Inferolator uses 1 shrinkage parameter for each gene, or about 6,000 total parameters. We also weighted differential expression evidence heavily. Inferolator uses it only as a filter on the regression results, and Genie3 doesn't use it at all. Finally, we weighted evidence of activation of a target when it was supported by both differential expression and co-expression more heavily than evidence of repression, reflecting the fact that eukaryotic organisms generally have more activators than repressors. In order to see if we could have an effect on practical genomics projects, we decided to use net profit to drive such a project involving a pathogenic fungus called Cryptococcus neoformans. Cryptococcus has a polysaccharide capsule, and when it lives out in the environment, the polysaccharide capsule is relatively small. But when it invades a mammalian host, the polysaccharide capsule grows large, and that growth is required for survival in the host and maintenance of the disease. So we used net profit to map the transcription factor network that regulates capsule size in Cryptococcus. The workflow starts with transcription factors that are suspected of having a capsule-related phenotype, and a few were known before we started. We deleted these transcription factors and then carried out RNA-seq, and then we fed the expression profiles into net profit. At the same time, we measured the capsule size of the deletion strains for each transcription factor. We took the network produced by net profit, and we analyzed, in the same way that I just described to you for the yeast genes, we analyzed the enrichment of net profit predicted targets of each transcription factor for genes that were known to be involved in capsule in some way, including transcription factors that we had previously identified through earlier steps of this process. Those transcription factors whose targets were most enriched for genes involved in capsule were then deleted again, and the process was repeated. We call this method of identifying predicted capsule-related genes phenoprofit, and it predicted 10 top transcription factors, of which 7 proved experimentally to have capsule-size phenotypes. Now, for comparison, there have been several previous mutagenesis screens, both random mutagenesis and directed mutagenesis, that identified 13 other transcription factors involved in capsule out of a total of 219 transcription factors. We also carried out validation experiments on the net profit network in Cryptococcus by chip-seq and by analysis of de novo inference of transcription factor binding motifs. And you can hear more about that by going to poster 34 at lunchtime. And you can also hear about another application of net profit to increasing the ethanol yield of fermentations with Saccharomyces at poster 44. I'd like to finish by thanking Brian Haynes, who was the primary developer of net profit, Zeke Mayer, who's the lead on the Cryptococcus project, Patricia Wang and Michael Kramer, who assisted with various analyses, and Holly Brown, who carried out the follow-up experiments on CBF1 and EDS1. Our work on Cryptococcus is in collaboration with the Tamara Deering Lab, and funding was provided by NHGRI, NIGMS, and NIAID. Thank you.
Questions? So I actually have a question. Is most of your original analysis, like the comparison with ChIP-ChIP, is that Rick Young's original ChIP-ChIP data set from way back?
No, it's all ChIP-ChIP experiments and ChIP-seq experiments on yeast as determined by two databases that try to be comprehensive. So it's 30,000 ChIP relations. If
you look specifically at ChIP-exo, is it true that the motif enrichment of your method is better than like the best?
I doubt that there's enough ChIP-exo data on Saccharomyces to do that, but I will look into it.
Yeah. More questions?
Hi, I have a similar question. So how do you, what do you think explains the higher performance by the PWM rather than ChIP? Because ChIP is considered as a true measurement of in vivo. Is it because you took a more stringent cutoff for the ChIP, or?
No, it's not because of a more stringent cutoff, because we actually, well, the cutoffs were determined by the authors. So we used all of the significant ChIP hits that were submitted to the database. Why do I think it's doing better? I think it's doing better because ChIP detects a lot of nonspecific binding that primarily is not involved in regulation of the targets.
Okay, thank you.
It's a good work, and I like it, but I don't understand how does net profit differentiates between direct and indirect regulation here.
So there isn't an explicit method of differentiating between direct and indirect, but it's implicit in the fact that we globally, our ranking is largely determined by the p-value for differential expression of the target gene when the transcription factor is perturbed. And what we find empirically is that those p-values are, the greater p-values tend to be direct targets.
Yep, I understand. Thank you.
So going back to your sort of original challenge of kind of moving beyond, do you think, I mean, a lot of the methods developed, especially in this community, train on one type of data and then validate against the other. And even here, you can show it again. Do you think we should, I mean, if the goal is more towards practical applications to genomics, how much better could we be doing by just being more opportunistic about using all the data together and then validating in terms of, maybe just some external biological results?
That's a good question. I mean, it's a bit of a dance in these projects in which you try to satisfy two demands. One is that you want to make prospective predictions that are completely independent of the data to prove that you haven't overtrained or anything like that. On the other hand, as you say, you want to use all of the data that's available to make the best possible predictions. So what we typically do is both. We first do either a cross-validation or a training on one type and testing on the other type. And then when we really want to make some expensive decision based on the data, we combine everything and get the best possible model.
Right. It was a great idea to couple the shrinkage parameter across regressions and also to couple experimental design to lasso. I have a quick question about comparison to these other algorithms. A lot of the algorithms that have been developed in more recent years tend to be more broad and don't focus on specific experimental design as much, because a lot of the data that people use tend to be heterogeneous and not necessarily around a specific design. So let's say around inherent variability in the population.
Could you say that more slowly, please?
So say a lot of the more recent data tend to exploit variability in inherent variability in the population, for instance. But some of the older algorithms, in particular, for instance, NIR, network inference by multiple regression, did take advantage of explicit experimental design in a manner similar to yours, although it didn't regularize using lasso, but by ridge regression instead. And it might be interesting to compare to such, unfortunately, an archaic method, but to see to what extent it's lasso versus coupling to experimental design that is driving the performance. Because I think some of the more recent algorithms are not so closely coupled to experimental designs anymore, which you pointed out.
That's a good point. I think by coupling to experimental design, you mean using the causal information from the perturbation of the transcription factor. Yeah, I will look into that, and I have to say the strongest conclusion from all of this work is really that is the key determinant.
Okay, quick last question.
Could the improvement in performance be to gene length bias?
Say that again?
Could the improvement in performance of net profit be to gene length bias? Because it's easier to detect differential expression with longer genes, and they're also more likely to have any of these motifs, binding motifs.
I'm sorry, I can't understand what you're saying. Maybe stand a little closer to the microphone.
So you show that net profit gives an improved performance, but you get that by matching the predicted list of genes to these binding motifs. And with RNA-seq, it's a known fact. It's been published in genome biology that it's easier to identify a differential expression with longer genes. The longer a gene, the more likely it is that it's going to be differentially expressed.
Longer genes are more likely to show up
Yes.
as differentially expressed.
And they're
yes.
also more likely to harbor these binding sites. So you.
Maybe we have to take the
Okay.
issue of bias offline. Just
I will follow up with you after the session. Thank you.
Okay.
Thanks.